Hello, Professor Stefero here, and today's video is on prokaryotes. This seemingly invisible but abundant group of organisms are the things that we probably often called germs. And germs are things that we have learned to protect ourselves from by using disinfectants, by wiping down grocery carts with disinfectant wipes, by coughing in the right way. All of these are ways that we have learned to protect ourselves from germs because certain prokaryotic organisms carry disease. But it's not true of all of them. Only a few carry disease, many are benign, and many are also beneficial. So this video is going to talk about characteristics of prokaryotes. We're going to look at their structure. We'll take a look at how they cause disease, but we'll also identify how they can be beneficial. Let's start by looking at the three domains. The three domains of biological life are Eukaryota, bacteria, and Archaea. And the two domains, bacteria and Archaea, are the ones that hold the prokaryotic cells. And that's the one we're going to focus on today. We see a variety of prokaryotes here, and they can also live in a variety of environments. They can live at different temperatures, at different pH, whether that be acidic or basic. They can live at salinity, so some prokaryotes live in fresh water, some live in the oceans or in saltwater conditions. Some can live without oxygen. Some have to have oxygen to survive. Some are photosynthetic. So it is a very diverse group using lots of ways to get energy and to transform that energy. Here we can see the scale of size of prokaryotes. They are very small organisms. So let's take a look at things that we know. We know that a human is somewhere between 1 to 2 meters, while the chicken egg is about 10 centimeters. Then we have the frog egg, and it's 1 millimeter. Then we come down to the bacteria, and they are 1 micrometer to 100 nanometers in size. And the 100 nanometer bacteria, the smallest that we have, require the use of an electron microscope in order to visualize them. Here's a pin tip. On the pin tip, we can see numerous bacteria stained orange. So again, this is an electron micrograph showing the tip of perhaps this pin itself and identifying how small bacteria really are. So let's take a look at some statements that helps us characterize the abundance of prokaryotes, but also identifies how small they are as well. So the first one here is about the mouth. More prokaryotes live in your mouth than the total number of humans that have ever lived. There are 10 times as many prokaryotes living in and on your body as the number of cells that make up your body. There are more bacteria that live and work in 1 linear centimeter of your lower colon than all humans who have ever lived on Earth. And last, the collective biomass, that is the amount of biological mass on the Earth, of prokaryotes is going to be at least 10 times greater than the biomass of eukaryotic cells. So we kind of tend to think of eukaryotic cells being more prevalent, more important, but the reality is that prokaryotes, even though they're invisible, they are by far the most abundant and most dominant life form that we have on Earth. Let's take a look at the structure of these cells. First, we're going to take a look at the different kinds of shapes that they can take. There are 3 shapes: cocci, bacilli, and spirochete. The cocci here are the spherical shaped bacteria, and these bacteria, an example would be Streptococcus. The next are the bacilli, and the bacilli are more rod-shaped structures. And an example of a bacilli would be E. coli. And then finally, the third shape is the spirilla and the spirochete. And the spirochetes and spirilla are corkscrew-shaped cells. An example of a spirochete would be syphilis. But whether they are cocci, bacilli, or spirochetes, prokaryotes share some similar structures. So let's look at some of those anatomical parts. First are ribosomes and plasma membrane. Ribosomes are small little particles that we could see here residing inside of the cell. These are going to be used to help make proteins for the cell. The plasma membrane is the outer boundary surrounding the cell contents. Next is the nucleoid. This is where the DNA or the chromosome is located in the prokaryotic cell. And here we can see the material in blue in the cartoon of this prokaryotic cell. And over here in the electron micrograph, we can see the nucleoid region with the DNA here. So this is a defined region, not a defined organelle like we see in eukaryotes. Another distinct characteristic is the presence of cell walls. Cell walls surround the plasma membrane of prokaryotes, and they are different than those that we talked about with regard to plant cells. Plant cells walls are made up of cellulose, but the cell wall of a prokaryote is made up of a combination of polysaccharides and polypeptide chains. It's called peptidoglycan. And the amount of this substance in the cell wall is one of the ways that we distinguish types of bacteria from each other. The capsule is another structure that's associated with prokaryotes. The capsule here is a sticky substance that's found on the outside of the cell wall. Capsules are important because they allow the bacteria to adhere to surfaces. And in this example, we see Streptococcus attached to this tonsil and probably giving this person strep throat. Flagella, Fimbriae, and Pili are the next structures. Flagella are the structures that allow the prokaryote to move through their watery environment. They kind of look like a propeller and work like a propeller as well, moving the bacteria through water and other fluids. Fimbriae are structures that protrude out of the cell wall. They allow the bacteria to attach to surfaces like your teeth or rocks. Another kind of projection that's a little longer than the Fimbriae are Pili, and Pili or Pili are the structures that are used between bacteria to transport DNA from one cell to the next. Here we can see some examples of flagella attached to a prokaryote, and notice the Fimbriae here also projecting off of the surface of this cell. So these are all of the structures we just identified. Let's take a look at some other characteristics now. Bacteria are very good at reproducing themselves. They can reproduce themselves in a whole generation within hours. And the way that they do it is through a process called binary fission. Binary fission copies the DNA that's in the cell and then distributes it into two copies. So every bacteria is going to be an identical cell to the other. And they can reproduce themselves very, very quickly. The DNA inside of a bacteria is about 1/1000th of the amount of DNA that we see in eukaryotic cells. In this slide, the DNA has been allowed to be released. You can see all of this stringy-like substance here, and that would be the chromosome for this particular bacterium. There are also DNA components that are called plasmids, and there are a couple of plasmids here. These are small circular pieces of DNA that are also found in the bacterium, and we'll talk more about these when we get to the chapters on biotechnology and when we talk about transforming bacteria. So how do bacteria cause disease? We know that some do, and the way that they do it is either by producing a protein and or releasing cell protein parts. In either case, those proteins serve as a poison to the organism that leads to the disease. So here in the figure you can see the two kinds of poisons. Those are called exotoxins or they're called endotoxins. And the exotoxin is the first one that we want to take a look at. Here we can see that these are the type that the bacteria cell or the prokaryotic cell produces a protein and then releases that from the cell into its environment. The endotoxin takes pieces of itself and those pieces then become the poison, and these often get released when the cell is dying or being digested. So let's take a look at some examples of each. So Staphylococcus aureus is an example of a bacterium that is an exotoxin. This particular kind of bacteria is found on your skin, and it can lead to what's called a staph infection. And this particular staph infection example in the foot here has gone a bit too far, but we can also get staph infections in our respiratory tract. These are common places. But Staphylococcus aureus is a bacteria that would release a protein that becomes the poison. The example of endotoxin is Lyme disease. Here we see the spirochete bacteria. It would reside in the gut of the tick, and then when you got bit by the tick, it would transfer that piece of the bacteria to you, and that would lead to the infection. And characteristically, when you've been bitten by a tick, there's a bullseye rash that occurs. Other kinds of endotoxic bacteria would be salmonella, septic shock. Those are all examples of endotoxic poisons. Another one that's kind of interesting is Clostridium botulinum. And this particular bacteria, when it breaks down, releases a toxin that will paralyze muscles. It affects the nervous system, preventing the neuron to stimulate the muscle. And we have cultivated that endotoxin and created a substance called Botox. And used in very small amounts, Botox is injected into the very small muscles that surround the eye, and it essentially causes paralysis. The tension in the muscle is reduced, and that reduces any tension in the skin that the muscle attaches to. So you can see the before and after pictures here where the skin is wrinkled versus the skin lying flatter on the face. So they do cause disease, but more often prokaryotes are going to be benign or they're going to be beneficial. And you have lots of prokaryotes in your body. We talked about that 10-to-1 ratio. Here's a figure of bacteria stained in blue within the digestive tract, where they would be used to produce vitamins and break down food that you can no longer digest. Here they're found on the surface of the skin. We can see bacilli and cocci type bacteria on the surface of the skin here, working to clean up dead skin cells. So they are the trillion friends you didn't know you had. Archaea is the next group. These types of prokaryotes survive in extreme environments, and they are the most abundant life form that we have, and they are found mostly in the oceans, but they can also be found in the gut of a termite and or the rumen of a cow, breaking down cellulose. In this figure we see Archaea in salt marshes like we would find around the bay, and each color here would represent a different kind of Archaea prokaryote. Here's another example showing the geysers in Nevada, and again the color shows the different kinds of Archaea, but they can live in very hot temperatures, they can live in very acidic conditions, so they live in the extremes. So love bacteria, they're the most dominant form on life. Love microbiology, and remember, you're mostly made of microbes. So take care, and I'll see you soon. Bye.